Welcome back. In the last lesson, we learned that the human body contains hundreds of thousands of different proteins, much like a large city contains thousands and thousands of different buildings. In this lesson, we will dive into how proteins are built and how their structures determine their function. Please take a moment to review the learning objectives for this lesson. If we think about buildings again, even though their final forms can look quite different, what they all have in common is that they're constructed from a limited set of component parts, like wood, stone, metal, and glass. How these parts are arranged and attached to one another is what dictates their final structure. The same is true of proteins. What those parts are and how they drive formation of three-dimensional structures is the subject of the next video. Starting with a fully assembled protein, in this case human insulin, the protein hormone that helps us regulate blood sugar levels, let's deconstruct it and see how it's built. If we unravel insulin's three-dimensional architecture, we can see that it's actually two long chains composed of tiny building blocks called amino acids, 51 to be exact. All amino acids share a common chemical architecture or underlying scaffold. Attached to this common scaffold is a side chain that makes each amino acid unique. In all, there are 20 different amino acids, each having a different side chain. Their common architecture allows amino acids to connect to one another like pop beads and form long chains or polymers. A polymer is simply a long strand of repeating building blocks called monomers. In a pop bead chain, each pop bead is a monomer. When amino acid monomers pop together, they form strong peptide bonds. So a string of amino acids is often called a polypeptide. Which amino acids are present and the order in which they're arranged dictates how the chain will fold up into a three-dimensional shape. And it's this unique three-dimensional architecture that gives each protein its specific function. In humans, the median protein is a string of 375 amino acid monomers. So at 51 amino acids, insulin is on the small side. The largest human protein is titin, a key component of muscles. As its name implies, titin is gargantuan, consisting of a string of up to 35,000 amino acid monomers. Once they assume their 3D shapes, some proteins are ready to function all on their own. One example is human serum albumin, the most abundant protein in human blood plasma, constituting almost half of total serum protein. Made by the liver, serum albumin is a chain of 585 amino acids that fold up into a globular shape. Serum albumin performs many functions, including maintenance of osmotic pressure, acting to buffer serum pH, and transporting hormones, fatty acids, and other biological compounds around the body. Now, let's look inside one of these red blood cells. Here you can see that the scaffold that gives the red blood cell its shape is made up of many different proteins connected like Lego building blocks. Each of these proteins is one part of a multi-protein complex. Amazingly, most of these larger multi-protein complexes are able to self-assemble once all of the individual protein parts are available. Perhaps the best studied multi-protein complex is hemoglobin, the highly abundant protein in red blood cells that carries oxygen and gives them their red color. Hemoglobin contains 4 separate polypeptide chains, 2 chains of alpha globin and 2 chains of beta globin. When these 4 chains come together, they form binding sites for another molecule called heme, which provides the site for oxygen binding. Now that we understand the fundamentals of protein structure and function, we'll next learn about how each different cell type knows which proteins to make. Where do the instructions come from?